Hello and welcome to the Biochemistry for Health Sciences channel. In today's video, we shall see why are proteins able to carry out most of the functions for a cell. What allows them to do all of these different functions? So that's a question we're going to try to answer in today's video. In previous videos, we saw the typical structure of a protein. Okay, this is a structure of a typical protein. We talked about some of the important features of proteins that are found in our water part of our cells. So these are non-membrane proteins. Typical structure of a non-membrane protein. Now we mentioned that proteins carry out most of the cellular functions. So just to remind you, there are proteins that affect gene expression. These are nucleic acid binding proteins. Proteins called enzymes can increase the rates of chemical reactions. Signal transduction proteins are proteins that help the cells respond appropriately to its changing environment. Then in the cell, there is a lot of transport that happens, and most of this transport is carried out by transport proteins. Defense proteins such as antibodies and complements protect us against microbes. Structural proteins provide support to cells and tissues. And then we have a category that we call miscellaneous proteins, and these are proteins whose function do not fall into any of these categories. So for example, there's a protein called ferritin that binds and stores iron in the body. So to answer the question, why can proteins do so many different things for the cell? The first reason is that proteins can form many different shapes. So here, for example, is a protein that has this primary sequence, and let's say this has a shape, something like this. Now, if we just change one of these amino acids, suppose instead of a lysine here, let's say we replace a glutamic acid, this shape will change. Okay, so similarly, if you change another amino acid here, that protein shape will change into something different. So just by changing the primary sequence of a protein, we can make all kinds of shapes. And shapes are very important for functions. Okay, so Quirley's question, why is shape so important for function of a biomolecule such as a protein? Well, Quirley, let's do something. Go ahead and cut this piece of bread with a spoon. How do you feel? Quite a frustrating experience, isn't it? Well, why don't you try to make a slice of the bread with a fork? That's also not an efficient way of doing things, right? Aha! If you now use a knife, you do get your slice of your bread. So you can see the shape, even in everyday life, is very important for carrying out a certain function efficiently. So the same logic applies to biomolecules. Biomolecules must have a certain shape to carry out a certain function. So that is one reason why proteins are very successful in carrying out many different functions for the cell. The second reason why proteins can do so many different things for cells is because proteins are made up of amino acids. Okay, any one of these 20 amino acids. And if you look at the chemical structures of these amino acids, you see that they have a lot of different functional groups, a lot of chemistry, a lot of diverse chemistry. So for example, these amino acids may have amines. Amines are very reactive in water. They can form hydrogen bonding, and they can give you positive charges. Okay, so amines have these features that are very important for the structure and function of a biomolecule. Carboxyls can hydrogen bond. They can be made reactive, although in water they're not reactive, but they can easily be made reactive, and they can give negative charges to biomolecules. Then there is an amino acid called cysteine, CYS. It has a sulfhydryl group. Now sulfhydryls are very reactive in water, and sulfhydryls can also give you negative charges, and sulfhydryls can also give you a very strong covalent bond called disulfide. So basically two sulfhydryls can get oxidized to give you a disulfide, or it can get reduced to give you back the sulfhydryls. So these covalent bonds add extra stability to the protein, as well as allows the protein to react in many different ways. And then the amino acids that make up proteins also have the hydroxyl functional group. So here's a hydroxyl, and these hydroxyls are very good in hydrogen bonding, and they can also be made reactive under appropriate conditions. And then we have amino acids that have amides, and amides allow the protein to carry out a lot of hydrogen bonding. That's very important in making all kinds of shapes. So this is why proteins can do so many different functions for the cell. One is that proteins can form a variety of shapes, and the second reason is that proteins have a diverse chemistry because of the 20 amino acids that make up proteins. So let's look at the structure of a typical amino acid that makes up a protein. The amino acid contains an alpha carbon, the alpha carbon shown here, and attached to the alpha carbon is the amine group. That's why we call it amino. And then we have the carboxyl group. That's why we call it acid, because this is carboxylic acid. The third group is a hydrogen atom. So this is common in all of the 20 amino acids: alpha carbon, carboxyl, amine, and hydrogen. The fourth group, which is different from one amino acid to the other, is called the R group. So that's the typical structure of an amino acid. Amino acid. Okay, alpha carbon with amine, carboxyl, and then the third group is hydrogen, and the fourth group is called the R group. So let's look at some of the key features of these amino acids. So let's start off with glycine. Glycine, GLY, is the 3-letter abbreviation. G is the 1-letter abbreviation, and that structure is shown here. The R group is simply a hydrogen atom, and that makes it the smallest amino acid among the 20 that you find in proteins. Since there are only 3, 1, 2, and hydrogen, these two are the same. There are only 3 groups attached to the carbon, 3 different groups, not 4. This alpha carbon is not chiral. For chiral, you must have a carbon that has 4 different groups attached to the alpha carbon. Glycine is very important, and we'll talk about this in future videos. Glycine is required for the synthesis of heme. That's the organic compound that makes your blood red, what you find in hemoglobin and other proteins. Glycine is used to make nucleobases, your purine and pyrimidine nucleobases that you find in RNA and DNA. Glycine is necessary to make creatine, and creatine phosphate, we'll talk about it later, is a very important storage form of ATP or energy in muscles. Glycine is also used for the synthesis of the bile salt that we call glycocolate. Now, glycine is also very rich in collagen, certain types of collagen chains, because it's very, very small. Just the hydrogen atom is the R group. It allows the collagen to fold very tightly and to have very tight packing. Now, it's interesting to know that some of these amino acids are actually neurotransmitters. So glycine is a neurotransmitter. Glycine is also part of a very important compound in our body called glutathione. This is an antioxidant, very important antioxidant in our cells. Very, very important because our cells, and even outside the cells, we get a lot of free radicals, but glutathione is an antioxidant that helps destroy or neutralize these free radicals. Glycine is also used for making foreign chemicals, what we call xenobiotics, more water soluble. So if you had a drug, for example, the body, this is a foreign chemical, your liver will conjugate glycine to the drug. This will make the drug more water soluble, and anything that's more water soluble can be excreted out of the body very efficiently by kidneys. So glycine is one of the chemicals that the liver uses to make foreign chemicals more water soluble for their subsequent excretion. Alanine, ALA, or the 1-letter abbreviation A, is also a very small amino acid because the R group is simply a CH3. So if this was alanine here, this would be a CH3. So it's a very small R group, just like glycine. And that small size, once again, allows alanine to be part of collagen. It allows very, very tight folding and packing in collagen chains. And these collagen chains are very important in our connective tissue, something we'll talk about in future videos. Alanine is a substrate for a very important clinical enzyme called ALT, alanine aminotransferase. And we use this enzyme in a hospital very frequently to diagnose liver functioning or liver diseases. Later under metabolism, we'll look at fasting condition, what happens to metabolism when you are in early fast or late fasting conditions. Alanine is used to transport carbons as well as nitrogen to the liver during fasting conditions. And that's a very important transport that we'll talk about under metabolism. Proline is an interesting amino acid because you see your alpha carbon here, the hydrogen, the carboxyl, and then the amine, and then you see the R group. Let me use a different color. So you see the R group, but the R group coming out of the alpha carbon, but the R group comes back and joins to that amine, the alpha amine. So this forms a heterocyclic ring, and here the nitrogen is joined to two carbons, one carbon here, one carbon here. That gives it we call it a secondary amine, and therefore proline is a amino acid rather than a amino acid, because the amine is a secondary amine. And because of this, so here is let's say a protein chain joined to carboxyl. This is your protein, and then your amine here will be joined to another carboxyl, another amino acid. You can see the chain is going to change direction. So you can see the protein chain forming a sort of a bend or twist. So this presence of this amino, the secondary amine, whenever you see proline in a protein chain, you tend to see a bend or a kink. So this is, of course, once again, very good for collagen, which has a lot of twisted chains. In collagen, a lot of the proline is hydroxylated to form what is called hydroxyproline. Another important thing about proline is a lot of our body surfaces as well as fluids have what we call proline-rich proteins, or PRP. And these proteins have very good antimicrobial properties. Then we have the branched chain amino acids, and you can see that's valine, leucine, and isoleucine. You can see the carbon R group coming out and forming a branch. Once again, you see the branch right here, and you see the branching right here. So these are called branched chain amino acids, and all of these have R groups that are very hydrophobic. They don't like water. And one important thing about these branched chain amino acids clinically is that in the disease called MSUD, maple syrup urine disease, there is an enzyme that is involved in the metabolism of these amino acids, and that enzyme is defective. That leads to a variety of problems in the babies that have MSUD. MSUD is now part of the newborn screening program. The aromatic amino acids. These are phenylalanine, tyrosine. Phenylalanine is F, tyrosine is Y, and tryptophan is W. So all of these have the aromatic rings, aromatic amino acids. So these, as you know, aromatic groups do not like water. They are very hydrophobic. And they also, the aromatic rings absorb very well at 280 nanometers. And in many cases, when we have a pure protein sample, we want to know how much of that protein is present in the sample. We simply measure the absorbance at 280 nanometers and calculate the concentration of the protein from the absorbance. In the genetic disease called phenylketonuria, or PKU, there is a problem in the metabolism of the aromatic amino acid phenylalanine. And so PHC, phenylalanine, will accumulate in the cells, and then it may be converted to side products such as phenylpyruvate, and these chemicals accumulate and cause all kinds of problems, especially to the brain. So we have delayed development as well as mental retardation in the babies having this genetic disease. In the disease cystic fibrosis, most of the, about 70% of cases of cystic fibrosis, the phenylalanine at position 508, so remember we start from the N terminal, 1, 2, 3, and so on. So in this CFTR, this protein CFTR that is responsible for the transport of chloride, which we'll talk about in a future video. At position 508, the phenylalanine is missing. We call this change in the phenylalanine at position 508 of CFTR. We call this delta F508 because F is the one-letter abbreviation for phenylalanine. The amino acid tyrosine is a very important precursor for a variety of chemicals. Tyrosine is needed for synthesis of dopamine, norepinephrine, epinephrine. So these are all hormones or neurotransmitters, as well as the thyroid hormones T4, T3, and the skin pigment melanin. So tyrosine is a very important starting material for a variety of important, clinically important chemicals. The amino acid tryptophan, TRP, is required for the synthesis of serotonin, as well as the hormone melatonin, and even small amounts of a vitamin niacin in humans. Then we have the sulfur-containing amino acids, and these are methionine and cysteine. And we talked about cysteine because of its sulfhydryl group. It allows the formation of disulfides. And disulfides are found mostly in secreted proteins because these proteins have to face a potentially hazardous or harsh environment such as the bloodstream. So for example, albumin has 17 disulfide bonds. Albumin is a blood protein, has 17 disulfide bonds. Antibodies have disulfide bonds. Insulin, which is another secreted protein, has disulfide bonds. So these disulfide bonds give the protein extra stability to its shape. Cysteine plays a very important role in many enzymes, such as cysteine proteases. Proteases are enzymes that break other proteins down. So cysteine plays because it's a very reactive thiol group, and it plays a very important role in the function of many different enzymes. Because cysteine is very important in many enzymes, unfortunately heavy metals, especially mercury, reacts very strongly with thiols, with the sulfhydryl group, and that can inactivate the cysteine in these enzymes and therefore affect the function of the cell. So mercury is a poison because we don't have too many of these enzymes around, and it doesn't take too much of mercury to basically stop the functions of all of these enzymes. Cysteine is also a component of glutathione, the antioxidant that is very important inside our cells. We can use a drug called N-acetylcysteine, and this helps us restore glutathione levels, especially in our liver cells, in case we don't have enough glutathione. So NAC can be used to increase our intracellular antioxidant, glutathione. A few words about methionine. This is the first amino acid in all newly synthesized proteins. Okay, so when we look at protein synthesis, the first amino acid is always methionine. And then later in protein processing, the methionine may be removed. So at the end of processing, some proteins will have methionine as the first amino acid, while other proteins you would not see methionine because it has been removed. Now, methionine is necessary for making a very, very important chemical in our cell called S-adenosylmethionine, or SAM. SAM is one of the most important methylating agent, and we'll talk about this later under metabolism. But a lot of our biomolecules, such as X for example here, can be methylated or must be methylated. Okay, add a CH3 group. So proteins, DNA, lipids, a lot of biomolecules are methylated. So a very important chemical called SAM, which requires the amino acid methionine. Then we have what we call the basic amino acids. Lysine, because there's an additional amine. Okay, so besides this amine here, there's an additional amine. Similarly, there's an additional amine here besides the alpha amine. And similarly, once again, we have a different amine besides the alpha amine. So we have an additional amine, and amines are basic. So proteins that are rich in these amines, these amino acids, lysine, arginine, histidine, we call these proteins basic proteins. Basic proteins are not very common, but when you do see them, they play some very, very important role, and most of their functions is due to the fact that they have a lot of these extra amines, and amines can give you positive charges. So a lot of these basic proteins are rich in positive charges. One example of basic protein would be histones, and we'll look at this later. Histones are very important in regulating the gene expression. So histones play a very important role in regulating gene expression, and what happens there is their positive charges due to amino acids such as lysine can be acetylated to remove the positive charges, and then to bring back the positive charges they can be deacylated. And because of the positive charges, they allow the histones to bind to DNA and therefore block the gene. So they play an extremely important role in gene regulation. Lysine is also a very important amino acid in collagen, a variety of collagen chains. We'll talk about this in future videos, but several of these lysines are hydroxylated in collagen, and some of them can also be oxidized to form very unusual covalent crosslinks, such as lysinol-leucine between the collagen chains. And this cross-linking of collagen chains allow these collagens to give our tissues strength, extra strength. And we'll talk about these collagen as a structural protein in future videos. Now the very important chemical nitric oxide is produced from the basic amino acid arginine by the enzyme nitric oxide synthase. Once again, we'll talk about this in future videos. Histidine is a basic amino acid, and this ring here is called the imidazole ring. Imidazole ring. The interesting thing about this ring that it has a very appropriate chemistry feature that we'll talk about this in the future has a pK around 6.5, and that allows this ring to act either as an acid, that means you can donate a proton, or as a base, that means it can accept a proton. And acid-base catalysis is very, very important in certain enzymes. So histidine plays a very important role in the mechanisms of several enzymes. Histidine is also used for the synthesis of histamine. And again, we'll talk about histamine in future videos when we talk about allergies and other aspects of immunology, the biochemical aspects of immunology. Then we have the acidic amino acids, aspartic acid, D, glutamic acid, E. These have additional carboxyl groups. So here's the additional carboxyl besides the alpha carboxyl. Here's another carboxyl besides this alpha carboxyl. So carboxyls, this extra carboxyl group, because of this extra carboxyl, we call these amino acids, aspartic acid, glutamic acid, as acidic amino acids. These amino acids are required for the synthesis of many other biomolecules. So for example, aspartic acid is required for the synthesis of your purine and pyrimidine nucleobases that you find in nucleic acids. Glutamic acid is a neurotransmitter, very important neurotransmitter. So once again, amino acids can act as neurotransmitters. And then the neurotransmitter that's called gamma aminobutyric acid, or GABA, that is actually made from glutamic acid. And then we have the antioxidant glutathione. That's to make glutathione, you need glutamic acid. Okay, so glutathione is has three components: glutamic acid, cysteine, and glycine. Okay, so very important antioxidant that we'll talk about quite a bit in the future. Okay, then we have the alkyl amino acids. Alkyl amino acids. So you have serine, that has the alkyl group here. Threonine, you see the alkyl group, and tyrosine, that has the alkyl group. So 3 alkyl amino acids: serine, threonine, and tyrosine. Serine is a very important amino acid for enzymes that we call serine proteases. These are enzymes that work on other proteins. Examples of serine proteases include chymotrypsin, that's the enzyme that we need for digestion. Factor 10A is an enzyme used for blood clotting. And then we have complements such as C1s, is a serine protease that we use for our immune function. Now, these alkyl amino acids are also very important for a process that we call protein phosphorylation. So if you have a protein P here, and then you have, let's say, OH, whether this OH is on the serine or threonine or tyrosine, what happens is that the cell can attach a phosphate group. And so you have now the protein, let's say here's a protein now that has the phosphate attached to it. Okay, phosphate attached to it. And phosphate, you can see, has 2 negative charges. And these 2 negative charges are going to affect the protein shape. It's going to affect the protein shape. So the protein shape might change. And when the protein shape changes, the function also is modified. So protein phosphorylation is a very, very important way to control protein function. So we have enzymes called protein kinases, such as serine threonine kinase, as well as tyrosine kinase. These are enzymes that catalyze phosphorylation of proteins in order to regulate protein functions. Now, this is a very, very important area, clinically important area, and we'll have many videos talking about these phosphorylation reactions to control protein functions. And not surprising, because proteins do most of your cellular activities, and that in turn leads to our anatomy and physiology. Now, in some proteins, we have carbohydrates attached to our proteins. These proteins are called glycoproteins. So these carbohydrates are often attached to these alcohol amino acids, to the hydroxyl of the alcohol amino acids. So here you have your carbohydrate chain, and this is called the O-glycosidic linkage. The O-glycosidic linkage. So these carbohydrates are usually attached to serine as well as threonine. And finally, it's good to know that serine is required for making amphiphilic lipids that we talked about in the past. The last class of the amino acids are your amide amino acids. Amide because their R groups contain the amides, CONH, amide. So they have amide R groups. Some proteins that are glycoproteins can have carbohydrates attached to them through the amides, okay? And these are called N-glycosidic linkages. So here you have the N, the nitrogen that's involved in connecting to the carbohydrate. These are called N-glycosidic linkages, and these are linked to the amino acid asparagine, or ASN. The amino acid glutamine is also required for the synthesis of nucleobases. Under metabolism in late starvation, we'll talk about how glutamine carries excess nitrogen to the kidneys and the liver for subsequent excretion and for conversion to urea, which is also finally excreted by the kidneys. And under late starvation in metabolism, we'll also talk about how glutamine provides the carbons necessary to make new glucose. And this is very important during starvation, because glucose, we'll see under metabolism, is the most important fuel for energy generation in all our cells. Also good to keep in mind that in proteins called keratins, these are very common in our skin, as well as other proteins, you can get cross-linking between lysine and glutamine. And these cross-linking reactions are catalyzed by enzymes called transglutaminases. And these cross-links are very important for these proteins in providing our skin and other tissues with strength. So they're very important for the anatomy of our skin. Another clinically important piece of data in the disease Huntington, we have this protein called the Huntington protein, and this protein has excess glutamine, more than the normal amount. And the presence of this excess glutamine in Huntington is been associated with the death of neurons, which then leads to the signs and symptoms of the disease, Huntington disease. So this is basically an overview, some of the important points of these different classes of amino acids. And we'll talk more about amino acids in future videos. Okay, so until the next video, goodbye and have a nice day.